Good afternoon, everybody, or at least good afternoon here on the 15th of January. I'm Steve Murphy, the chair of CASIOPA, Center for Applied Sciences in Ontario Protected Areas. And today we are lucky to have Estella Crosby, who is going to do rapid understory restoration techniques, Cache Lake and Algonquin Provincial Park, which is always very nice. We have a nice little audience. Thank you, and welcome to all of you. And without further ado, you're going to see me disappear and shut up, and I will turn it over to Estella. Estella, take it away.
All right, sounds good. Thank you. We were having some mic issues, which is why I'm having to hold it so close to my face, but we will get through it. So yeah, hi everyone, and thank you for coming. I'm Estella, and I'm a master's candidate in Steve's lab, and this webinar is going to be about my master's research. I'm studying restoration techniques of a red pine plantation in Algonquin Provincial Park. I'd like to start with a land acknowledgement. The University of Waterloo, which is the institution I am studying at, is located on the traditional territory of the neutral Anishinaabe and Haudenosaunee people. The majority of Algonquin Provincial Park, which is where my research site is located on the unceded traditional territories of 10 Algonquin communities. Indigenous people are the original stewards of Turtle Island and have been here since time immemorial. As a settler, I'm grateful for the opportunity to live, work, and study on these lands. So before I get started talking about my specific project, I'd like to give a little bit of background context. The understory of a forest can be described as the ground level vegetation, and it has beneficial impacts on the forest ecosystem, such as increasing the overall vegetation diversity, providing more varied habitat for insects and animals, and increasing soil nutrients. In pine plantations, the understory is sparse to non-existent due to shading from the tree branches growing together, as well as the thick layer of fallen pine needles, which can suppress vegetation growth and acidify the soil. Pine plantations are prevalent in Algonquin as well as in Ontario and Canada. Due to the high number of these plantations, there has been interest in how to restore them. However, the time that projects take as well as their cost are major barriers to restoration. And because of this, this project tests understory restoration techniques that are both cost and time effective. So before I jump into research specifics, I'm also going to talk a little bit about the context and the history of my specific site. So it's located on the north shore of Cache Lake. If you can see, it's where the white marker on the Algonquin Provincial Park map is pointing to. It used to be home to a variety of buildings, including park headquarters, a luxury hotel, and a train station. These buildings were demolished in the late 1950s, and the area was left bare for several years. In the 1970s, a red pine plantation was established, and then the area was left unmanaged until several years ago when this project began. However, some remnants of these human disturbances still remain at the site today, including the hotel steps and pieces of the foundation. There's also a historical walking trail within the site still present, and close to the site, there's a parking lot and a boat launch. So this area still experiences human-caused disturbances. This research project is a 4-year partnership between the University of Waterloo and Algonquin Provincial Park, which spanned from 2021 to 2024. I'm in charge of the 3rd and 4th years of this project, and the first 2 years of this project were completed by my wonderful lab mate, Jason Phoenix. So, prior to the restoration treatments occurring, the experimental area was thinned and herbicide was applied to invasive species by park staff. The thinned trees were left at the site instead of being removed. This is because leaving dead wood at the site can benefit the ecosystem. As the wood decomposes, it can increase the nutrients in the forest floor. Additionally, some species of lichen and mosses are well suited to growing on dead wood, and some insects specialize in deadwood survival. Also, leaving the cut trees increases the overall heterogeneity of the area. As you saw on the previous slide, the research site is divided into 4 blocks. These blocks are further subdivided into an 8x8 grid, which are the treatment plots. Each plot is 8 meters by 8 meters. The treatments were assigned to individual plots using a randomized block design, and each plot is identified with a unique 3-digit code, with the first digit being the block number, the second digit being the row number, and the third digit being the column number. So there's a total of 5 different types of plots in this experiment. The first is the unthinned plot type, which I added. So this area received no restoration treatment, no thinning, or no herbicide. It's the pine plantation as it was before we got in there to restore it, and these plots are to act as a reference state for statistical comparison. The next plot type is applied nucleation. So applied nucleation is when trees or shrubs are planted scattered throughout an area instead of fully planting the area. This takes less time, money, and resources than fully planting, and also mimics natural succession more closely. Beaked hazel shrubs and yellow birch trees were used as the nuclei in the experiment, and each plot which received the applied nucleation treatment received either a beaked hazel shrub or a yellow birch tree, not both. The next plot type is called the wind-throw guard plot type. So the idea of a wind-throw guard is planting a herbaceous species in a ring around a woody species, and this is thought to reduce damage or mortality from the wind. Canada goldenrod was used as a wind-throw guard species in this treatment. So there are plot types which have just applied nucleation, plot types which just have wind-throw guard, so a ring of goldenrod, and then plots which have applied nucleation and wind-throw guard. So a beaked hazel shrub or a yellow birch tree surrounded by a ring of goldenrod. These plants were chosen because of easy access, as there were many growing near to the site, the fact that they're native, early successional, and light tolerant, as the understory will be receiving lots of light due to the tree thinning which occurred. All of these treatments, as well as the laying out of the experimental grid, was done by my predecessor in 2021 and 2022. So, all of this leads to my research questions, which are, which treatments are most effective at restoring understory plant and insect diversity and richness? Which treatments are most effective at restoring chemical soil parameters? And do wind-throw guards reduce the mortality of transplanted shrubs and trees? So next, we're going to talk about how I'm going to answer those questions. So the first method/data type is the soil data. Soil sampling occurred once a year every year, and it occurred in a W-shaped transect throughout the site and also in the unthinned plots. So the X's mark the W-shaped transect. This is due to feasibility, both the cost of testing the samples as well as the time it takes. Because there are so many plots, unfortunately, we were not able to sample every single one. So the samples were taken in 10 centimeters deep into the ground, and they were stored in a paper bag to help prevent mold growth, and then stored in a fridge once they were brought back to the lab at Waterloo. And they're being tested for phosphorus, nitrate, nitrogen, potassium, and pH. The next method/data type is the vegetation data. These also occurred in the W-shaped transect and the unthinned plots as time permitted. And as time permitted, I used a random number generator to pick additional plots to survey by randomly generating the 3-digit code associated with the plot. This was to prevent any bias in me preferentially choosing plots. These surveys occurred 4 times per year over the summer, so roughly once a month. A 1 meter squared quadrat was used, which was placed in the middle of each plot, again to reduce any bias by me preferentially picking an area which had maybe interesting looking vegetation. All of the vegetation species in the quadrat were identified to species, and the percentage of the ground they covered was estimated using the Dobenmeyer cover classes, which are in the table, which groups percent coverage into 6 easily recognizable and estimatable groups. The next method/data type is insects. This again occurred in the W-shaped transect due to the time the insect identification takes, and this occurred twice each summer. And I added this survey to the project, so it only occurred during the summers of 2023 and 2024. I used 2 different types of traps, both pitfall traps and pollinator traps. So the pitfall traps were dug and left in the ground with the lid on for at least a week to prevent bias, as the insects might explore something new in their habitat, and they were filled a quarter full with ethanol. The pollinator traps were filled with water, and a few drops of unscented detergent was used to break the surface tension, and unscented detergent was used so that as scented may attract insects from outside of the immediate area. When the pollinator traps were hung out, the lids were removed from the pitfall traps and they were both left out for 48 hours. Then they were collected and the insects were stored in 70% ethanol and brought back to the lab here at Waterloo, where I identified them to ideally family. The final method/data type is the mortality surveys. So this occurred in all of the plots other than the controlled and unthinned plot type, as those received no transplants, and this occurred one time a year. So any plots which received the Winthrop or goldenrod transplants, the number of goldenrod stems, if there were any, were counted and recorded, and if none were alive, a 0 was recorded. And for any plots which received the applied nucleation treatment type, whether or not the applied nucleation shrub/tree was alive or not was also recorded. As you can easily see in the middle picture and less easily in the other two pictures, all the applied nucleation transplants had a piece of flagging tape tied to them, so they could easily be identified as the applied nucleation treatment and not just a shrub or tree growing in the site. So all of that brings me to my very preliminary results, as I haven't had time to do a whole bunch of statistical tests, but we can talk about what I've seen so far. So for the vegetation surveys, the most commonly observed species was wild red raspberry, which had 208 total observations, which was followed closely by mayflowers, which had 204 total observations. And just to make clear, the vegetation observations are not the total number of plants observed, it's the total number of plots which contained at least one of these plants. The vegetation observations spanned the course of the project, so from 2021 to 2024. For the insect data, the most commonly observed family was hoverflies, which had 56 individuals caught in the traps, followed by ants, which had 53 individuals caught. And these observations span my portion of the project, so 2023 and 2024. For the mortality of the transplants, 83% of the applied nucleation transplants survived in the applied nucleation treatment, and 89% survived in the applied nucleation and Winthrop treatment. It should be noted that 4 years isn't super long in terms of the forest plant community or ecosystem recovery in general, and I also haven't had time to do much rigorous statistical testing, so none of this is definitive in terms of long-term outcomes. However, I would say that it's a good sign that so many of the transplants survived, and also that the most commonly observed vegetation species are native. Next, we're going to talk, or I'd like to end with some fieldwork mishaps and lesson learned. I think it's important that these be discussed as there are the reality of working in a field like ours, and sharing issues and solutions might make things easier for someone else down the line. So in no particular order, and this is not all of this issues I faced, but over the past 2 summers, but this is some of them. So the first is that the quadrat I was using for the vegetation surveys, the wood snapped multiple times. I think the wood was just not very thick and it also got very wet because it was pouring rain for several days. Luckily enough for me, the Algonquin Wildlife Research Station, which is where I was staying while I was doing my fieldwork, was able to lend me a much sturdier one that I was able to use for the rest of my vegetation surveys. Another issue I had was that when the insect traps were out at one time over the summer of 2024, it poured rain, and the pollinator traps, which were hung up, some of them got very full of water and fell onto the ground and tipped over. I did my best, I collected them and did my best to salvage the insects which were in the traps and identify them. Luckily, so far, although this is not definitive, it looks like there aren't really any crazy outliers from that set of trapping, so hopefully we should be able to use the data. If not, it may have to be cut or amended somehow to account for this. Speaking of insect traps, another issue I ran into was that sometimes slugs get into the insect traps, and during the first summer of my fieldwork for this project, I stored all the insects and slugs in 100% ethanol. However, some of the slugs kind of dissolved or rotted in the ethanol, which was both kind of gross and also contaminated some of my samples, so I wasn't able to use them. However, this summer I switched to storing everything in 70% ethanol, and that seems to have done the trick. And then two more. So another issue I ran into is that each plot was labeled with the 3-digit code written on with Sharpie on a piece of flagging tape. And this faded in the sun or with the rain, so that made fieldwork a lot harder because it was harder to figure out exactly what plot I was in. A solution we came up with was to use metal tags, which I could kind of engrave the plot number into, and then they couldn't fade. And last but not least, the vegetation surveys originally occurred in the entire 8 meter by 8 meter plot. However, doing these surveys took a very long time as I had to identify uh, every single plant to species. And also, I'm not the tallest person, so it was a bit hard for me to correctly estimate the percentage cover of a plant in an 8 meter squared area in a forest. So we changed it to a 1 meter squared area, which meant that the vegetation surveys were able to go quicker, and I was also able to get more done. So that's all I have today. I believe we have time for questions, but if we don't get to yours or if you think of something later, my contact information is up on the screen. Thank you so much.
We sure do. We have time for questions. And again, we won't badge you, Estelle, or anything like that. We have lots of folks on this call who are very pragmatic people like you. And as you may guess, we set this entire project up to, do good science. But a great deal of it was also to think about sort of the practicalities of doing some of these fairly decent scale restoration work as well. Um, so that's the reason why Estelle is emphasizing it. And I would concur, Estelle, if I may editorialize briefly, the survival rates over 4 years of the particular plants is quite good. Like, it's better than I usually see for myself, and I've been doing this for a long time, right? So I think you and Jason have done a great job of doing it collectively in terms of what goes on. That's a pretty impressive number. And goldenrods, weirdly enough, actually, you might think they're really robust, but they sometimes are really persnickety. They don't always survive transplants quite well. That's enough for me from now. Certainly, I'll flow the floor open for any questions, discussion. It could be, if you prefer, actually, as opposed to questions, but people are more than welcome to weigh in. If not, that's fine. I don't, want to press anybody, but we'll just go from there. Oh, Aaron, go right ahead, my friend.
Hi. Yeah, I'm just interested if there was any consideration to burning in terms of restoration of red pine. I know the Muskoka Watershed Council is, or no, sorry, Friends of Muskoka Watershed is working on an ash project in the Muskoka region where they're adding wood ash to increase phosphorus and calcium in forests. And I was just wondering if that was considered or if there was, an ability to add this in future years.
I think I personally wasn't super involved in. Oh, no, I'm not on mute. I personally wasn't super involved in the planning of this project. I just kind of took it over, so I might let Steve take this one.
Yeah, Aaron, it's a very good question, and fair enough. I mean, so you weren't involved in it. You weren't even here as a grad student at the time. Um, we discussed a whole bunch of options, and there's always a bit of a fear within the provincial park system of using prescribed burning, although I don't suggest that they were completely reluctant to it or anything like that. A lot of it just came down to the practicality. They were kind of like, when was this going to occur? They wanted to thin it. That was kind of the sort of the directive from the park itself, which I don't disagree with. I don't think it precludes the possibility of doing some burning later on this or related sites, and it's certainly something that's been under discussion kind of on and off. So I think your insight is well served there, and I suspect actually that a lot of it was just because the decision was made based upon what they could do or what they gave permission to do at the time in the park, and that was passed on to me and then Estella and Jason as well. But I do think there's some opportunity to think about doing exactly what you suggest, that burning could be an option, especially for some of the areas which are even close to the lakes, because there's a little less risk there. So I think it's a great suggestion.
Yeah, and just to follow up there, the wood ash applications that they have been doing is just putting wood ash on the forest floor with some pretty remarkable early results. I can't remember what university they worked with, but there was something they did it on some sugar bushes, and there was something like 20 or 30% increase
Mm-hmm.
in like phosphorus and calcium and stuff in the tree within like 4 months of the application. So
Yeah.
that may
I,
be something to consider as well.
I think the one you're referring to and I could be mistaken because there's lots of these little side projects that we've been involved into. I think that was the one that was primarily University of Toronto when Basilico was there. He's now gone a couple places now. He's at Lakehead. And one of my grad students, Michael McTavish, helped out with some of these projects. So as not as his thesis, but as a side project. So that might be the one you're referring to. I'll have to ask Michael. Actually, I'll ask Michael because I mean I don't try to know some of the people's business. He of course came to me and asked, can you do it? I think that's one of the ones he was working on actually, but I wasn't involved directly in it. Obviously, I think they asked me a few questions, so I'm sort of thinking, well, maybe that's the same one. So it'd be kind of interesting to see if that was the intersection of that one you've talked about. So yeah, no thanks. That's a really good idea in terms of sort of bringing it up, but also I did remember. I think with things like that, they've had, as you as you said, remarkable results about how this done. This is good. Any follow-up questions? Any follow-up discussion from anybody is more than welcome.
There was a question in the chat from Damien,
Oh,
and he
go ahead,
asked
please.
me
Yeah, I
if
know my chat up.
I've
Sorry.
no, it's okay. He asked me if I've ID'd the slugs, and he said he would assume most of them are non-native. I have not ID'd the slugs because most of them were kind of dissolved. But to be honest, I don't know enough about slugs specifically to comment. But it did look to me like they all kind of looked the same. So I would assume they would be all the same species or at least the same family or group. But I can't comment further on that right now.
I will, and Damien, you're welcome to chime in, of course, unmute yourself and get in, get into the chat, into the verbal here. I think Damien's right. They're probably going to be the big non-native one in Ontario. I think that's entirely correct. I think they are exactly as you describe it. We'll take a look at it, but it wouldn't surprise me. The ones that I pick up, for example, down here in Waterloo, almost always are the non-native ones. So yeah, I agree with you. Something to look out for, though, Estelle. I think Damien's advice is a good one. We should sort of keep an eye on that and sort of say it'd be a little more remarkable, actually, to use that word again, if they weren't non-native. I think that'd be kind of more interesting. But I think they're likely to be the invasive ones, unfortunately.
Yeah, I'll definitely look into that. Thanks for the tip.
Yeah, thanks, Damien. That's very helpful. I hadn't really twigged onto it, but it's like, yeah, we should really pay attention to that. Yes, absolutely. Be interesting if they weren't though, being in Algonquin. You never know. Maybe it's different. So. Other questions, follow-up discussion by all means. I won't keep people past the half an hour. I do respect everybody's time. Appreciate people being here live. And Estelle's time, of course, as well. And Estelle, I promise not to sandbag you. I don't have any questions. I get to ask you enough questions as it is. So
No, that's okay. Damien asked a question, fun question. What's your favorite species observed in the study? Red pine, yellow birch, and beaked hazelnut are some of my absolute favorites. Yeah, those are good ones. I think my favorite species as I've observed is the pink lady slipper orchids, just
mm,
because I didn't really know that much about them,
mm,
and the fact that there's just orchids growing in a pine plantation was pretty
they do.
crazy, and they're very pretty. So, yeah.
Yeah, that's I remember the first time I looked closely at the pine plantation in Nova Scotia. There's another orchid species that grows there as well, actually related. And Estelle, my reaction was the same as like I knew enough about plants as like wow, that's great. And it's like, why isn't a plant plantation? I realized, oh, it's acidic. So, but I figured the duff would be too high for it, right? I mean, it'd be too much layer, but that's it. So, Damien, I appreciate that actually in there. And Damien, for your questions, it's in the chat actually. I actually very much like yellow birch. A lot of people, for some reason, kind of dismiss it, but I actually like yellow birch a lot as well. Actually, it's a resilient species, which is kind of cool. So it's all good. Anything else anybody wants to ask? Fun or not, or maybe not fun, but other than that.
Did you get any rattlesnake plantains at all in
Mm,
there? They're another woodland orchid species that I see a lot out on Georgian Bay, in pine forests. So I'm
mhm.
interested to see if you guys got any of those.
No, I haven't. I didn't get any rattlesnake plantains. No.
That's true. I have seen them too, actually, as well. And Bruce in the Bruce Peninsula, you're actually right about sort of what goes on. Goodyeria is kind of an interesting little one, actually, at the end of the day. So it's kind of neat. Um, yeah, it's kind of funny in there. Goodyeria is kind of a pretty common one, actually. So I'm surprised we didn't see it. But then I don't know. I mean, Algonquin's not maybe the best place for it, but I have seen it in northern Quebec, so you never know. Good question. Anything else people would like to ask? I kind of like the whole natural history angle we've gotten into too. It's really fun. I always like natural history. All right, seeing none for now. I'm going to again thank Stella. Thank you very much for doing this. Much appreciated. This will, of course, go on in the YouTube channel, so it'll be posted within a day or so. So it should be up there if anybody wants to sort of see and review it. Thank you very much for all of our guests today for coming in and seeing it. We really appreciate it. And you'll see some fall from there. And Stella, of course, did leave her contact information. You can always get her hold of her through me as well, but she's the expert on this sort of thing. I helped design the project, but she's the expert. She did all the real work actually out of there. So it's all good. Well, thanks again, once again. And I will drop off and conclude the meeting. Thank you all for coming. Much appreciated. Thanks,
Yeah,
Attila.
thanks everyone for coming.
Bye bye.
Thanks.
Bye.